Can everyone see now?
Yes.
Yeah.
Yes.
Jolly good. Right. Here's the talk. pH. I'm going to give you a bit of a talk about basically what it is and how one would measure it. I'll just get rid of something there. Oops, sorry. Oops. Just 1 minute. Just bear with me 1 minute. Just get rid of that. Yeah, that's better. Yeah. Right, here we go. Right. Basically, pH matters, but it doesn't matter too much. I suppose what I mean to say is people shouldn't get bogged down with worrying too much about measuring pH in the first place, but it does have a huge effect on the brewing process. I don't have a huge knowledge of the chemistry involved in some of the processes, so you'll have to bear with me on that. Do feel free to interrupt and ask any questions as we go along. Basically, I'm going to break this down into different sections. These sections will be what pH means, why you would measure it, a bit of history on the measurement of pH, different measures and measurements, buying a meter, calibrating it, and care and maintenance of your meter. What it means and why one would measure it. I'm breaking this bit down into a little bit about acids, bases, some talk about acids and alkalis, or acids and bases as they're also known, hydrogen ion concentration and how it affects pH affects the brewing process. I'm not going to go into a huge amount about the pH and the brewing process. That's a whole talk in itself, really. Possibly tempted to do one at a later date on that. Acids and bases. Well, what you've got here is you've got basically a pH scale which goes from 0 to 14. 7 being in the middle is your neutral point. You've got your acids where there's an excess of hydrogen ions. You've got your bases, which basically are where you've got hydroxide ions. The neutral point is where there's a balance of these two things. The pH stands for, well, it's not exactly known what pH truly stands for originally, but has become to be known as the potential hydrogen. So it's the potential for hydrogen ions. This scale is logarithmic. What this means is that for every change in 1 unit, you're changing 10 times the amount. So basically, from pH of neutral 7 to 6 would be 1 unit, so to speak, but then from 6 down to 5 would be 10 times that, 5 down to that, 10 times that, and so on. So it's a scale that is a little bit like a couple of other scales, the decibel scale and so on, where logarithm is used. What it shows, though, is that the difference between neutral and slightly acidic or slightly alkaline is very slight. What one will notice is that trying to measure the pH of distilled or RO water, which should come out at around 7, any slight pickup of any chemicals will push that over to being acidic or alkaline. Right. On this picture, you can see here some common pHs of various items. You can see the classic hydrochloric acid down there at a pH of 0. Having said that, there are some acids which are below 0, just in this odd way that when they put the scale together, um, there are also alkalis that are stronger than 14. Here's another picture with some more common items showing that seawater is slightly alkaline. Um, Water itself will pick up carbon dioxide, which will tends to change the pH and make it slightly acidic. So when measuring, again, when measuring pure water, carbon dioxide will make a difference. You can see your liquid drain cleaner right down there at 14, along with bleaches and the sort of soaps and so on, which will be your alkalines. Um, Here are some pH levels of finished beer. You can see there sour beer down at 3, and pale ales and so on up at the 5.5s, 5.3s, and so on. But they're all within this range, typically. So when we're measuring pH, we're usually looking at measuring pHs within these ranges, which is relevant to when you're using a pH meter. Here's another diagram of the um, hydrogen ion concentration. In other words, what we're actually measuring when we use a pH meter. You can see that obviously neutral, this pH 7 in the middle, you've got a balance of hydrogen and OH ions, which is your neutral point. And you could see how the logarithmic effect of this is shown by this picture here. And again, at the bottom of the graph there, you've got some common items like star sand, the beer, as it would be finished, the mash range roughly, and then drinking water and so on. Right, pH and the brewing process. This is really why you'd actually bother to measure pH in the first place, to check on some of these things. Probably the most important one is enzymatic activity. In other words, during the mash, we've got enzymes that are, they have an optimum pH for their activity. Now, these optimum pHs, they're not set points. A lot of enzymes will work within a range before they would actually get denatured, but their activity varies, and there obviously is a sweet point for the pH of a particular enzyme. Different enzymes are going to have different preferred pH value, well, the ideal pH values. So the brewing process tends to be more of a compromise to finding the best extraction, and in other words, the best enzymatic activity, therefore the best extraction. You've probably got basically for mashing, you've got a pH range of somewhere between 5.2 and 5.7, average of around 5.5. You could push that lower. There are people who claim that it's better at slightly lower pHs than those. The tannin extraction is also an important consequence of pH levels. Tannins are going to be produced if you're at a pH of above, say, something like 6. So you might find yourself, say, acidifying your sparge water to reduce tannin extraction. And there's another place where you might be wanting to just check your pH. Although, as I said before, checking the pH of water is tricky because it doesn't take a lot for it to be pushed into either acidic or alkaline, just because at the neutral point, we are talking about very subtle differences. Protein coagulation is another thing affected by pH. Ideal at around 4.9, but again, because of other aspects of the brewing process, one wouldn't necessarily go for 4.9, but that is something that's relevant there. Hop utilization, again, hops are utilized better at a higher pH. In fact, a lot of hops utilization is probably best at about 7, 8, or even 9 pH. But obviously, we don't go there. But again, something worth being aware of. Maillard
Jerome.
reactions, another thing that's. Oh,
Sorry,
sorry,
Jerome.
go on, yes.
Jerome, sorry, you're saying the higher pH is better for hop extraction. Is that in the kettle or in the fermenter, or and
Oh,
also,
in the kettle. In the
and
kettle, yes.
and why is that?
I think it's due to the isomerization of the oils and so on are better at a higher pH. So as far as I am aware, it's the extraction of those hop oils works better at a higher pH.
Okay, thanks.
Again, Maillard reactions, that's another thing that are affected by pH. They work better at a higher pH again, which is why some of the processes for decoction and so on mention methods to keep the pH higher at certain stages. We've got yeast nutrition as well, another place where the pH is vital and has a huge effect on the process. Yeasts themselves will actually affect the pH. Different yeasts will affect the pH differently. And what you've got with the yeasts is that the yeast has a proton pump, which basically lowers the pH in order for the yeast to get a higher pH. So the yeast inside the yeast cells wants to be at a higher pH. And by using a system, it will end up by lowering the pH of the beer itself, which is those that if you actually follow the pH of a fermentation, you'll find that it rises slightly at the end. And this is where the yeast is autolyzing, it's dying, and therefore the pH slightly rises as those dead yeast cells cease to be doing their action and of keeping the pH lower. Well, spoilage. Here's another thing where basically by keeping the beer at a low pH, you are going to keep the beer better. Basically, anything above 4.6, I think 4.6 pH is what the US Food and Drug Administration uses their upper limit for packaged beer. And basically, again, one might want to be looking at measuring the pH of finished beer for that reason. And then you've got finings. Finings are affected a lot by the pH of the beer. The coagulation and dropping out and so on will all be affected by the pH. And at the end, taste. there are claims that a lower pH in a beer produces a crisper, brighter sort of taste to the beer, rather than a more muddied taste when pH is higher.
So sorry, Jerome, for finings, which way helps the finings more, a lower or a higher pH?
I think it's a lower pH again. I think I'll check on that. Having said that, it affects this is only really about isinglass and so on, the collagen type ones. Irish moss, I think, acts differently, and it's to do with the isoelectric point, I think, of um the yeast. Well, I think it's to do with something to do with the charges on the yeasts and the proteins and so on. So as far as I know, it's by them being charged at a certain charge, and that is enhanced by certain pH levels. I think it's that the lower pH is better for dropping out.
Okay, thanks.
Here's just a quick classic diagram of the what goes on with enzymes in the mash, and basically showing pH at the bottom there, the ideal range, pH range for certain enzymes to be active at. And again, it's got the temperature on there as well, showing these areas in which enzymes are best active, and showing that pH affects these quite considerably. Little bit of history here. Just going to put this into early indicators, bit about Sorensen and a bit about Beckmann. Early indicators, this is a jar of litmus, which was the first sort of indicator to be sort of commonly used. These indicators were originally used for processes such as dyeing, where knowing whether a solution was acid or alkali would then allow you to use particular dyes and so on. I think it was sometimes in the 1300s, a Spanish scholar by the name of Arnaldus de Villanova started using litmus first of all, and it's extracted from various lichens. So the litmus, as in litmus paper and the litmus test, is a lichen-based, various other plant materials, but mostly certain types of lichen, which will change color according to whether something's acid or alkali. This is probably the major thing about pH, and the history of pH. Sorensen, who worked for the Carlsberg Brewery Laboratory, yeah, basically he devised the pH scale in 1909. He was working in the laboratory on protein, the study of proteins and how they affected the mash, and was trying to find a way of quantifying the acidity and alkalinity of the process. Him and his wife, in fact, who was very involved as well in the laboratory. This is the Carlsberg laboratory that basically came up with the Carlsberg yeast, very important in the history of beer and brewing, and is why we have the pH scale today, is that classically down to brewing. He devised this scale in 1909. It was quite difficult to measure initially. It used quite a lot of complicated laboratory techniques, but it was down to him that we had the pH scale. In 1934, Beckmann came up with the pH meter. Now, this was in response to the fruit growers in America who needed to have a means of measuring the acidity of their fruit juices, and other methods such as using litmus wouldn't work because of the effect of some of the constituents of the juice, which would affect the paper and so on that they were using at the time. So he came up with the first proper portable pH meter. He called it the acid meter first of all, and then was changed to the pH meter. Beckman then went later, gave up his job at Caltech and set up a company making these meters because they were so successful. And I think Beckman pH meters were certainly available up until recently. Right, we got onto different methods of measurement of pH. I'll start with the classic cabbage. Basically, you can make an indicator yourself that will show a scale somewhat like that, and you can make it out of basically boiling down cabbage. Here we go. You thoroughly chop a cabbage, boil it, strain it, and then you use this juice as an indicator. And this is because of anthocyanin pigments that are in the juice, which change very subtly according to a change in pH. Having said that, it only works within a certain range, and is not really, apart from a bit of fun, not very useful, because the subtle changes, there changes over at least 1 point or whatever, and subtle here and there. So we can use indicator papers to measure pH. Now, they're cheap, it's easy to use. You just dip them into your solution and then compare the color that they go to the color on the color chart. But they're not very accurate. Now you can get what they call narrow range papers, which will work within 0.2, 0.3 of a pH unit. But that's as good as you're going to get. Some of them you have to be careful that they can be difficult to read if the color of your sample is such that that might affect the paper as well. So there is that slight difficulty. They are good though for doing basic checks just to show that you're in a sort of right ballpark, so to speak. They're excellent for checking your star sand, for instance, just to check that that's still in the right place. There's another picture of the color chart that you get on pH papers. Or you can go for the pH meter. They're an electrical device which basically measures a potential difference, so basically a voltage. Now, they're basically what all modern pH meters these days, nearly all of them will be what they call a combination electrode, where you've got a reference electrode and your so-called sample electrodes together in one package. And so they'll be called combination electrodes. They do require calibration, and I'll go into that shortly. Here's a typical pH probe and how it's built. We'll start at the bottom, right, and basically you've got this glass bulb at the bottom there, which has a very sensitive coating on it, and this glass bulb is permeable. They're very, very thin. They are very delicate, so one has to look after this bit particularly well. I'll talk a little bit later on about care and maintenance, but you've got this bulb at the bottom. Now, ions are going to be leaching through this into the solution inside where there's an electrode. This ion exchange will be amplified by some electronics, and then that signal will be displayed. But you've got a reference electrode as well, and the reference electrode allows the measured solution to be that voltage to be against a reference voltage in a solution that's, you can see there in the side part of the electrode. So you've basically got an electrode within an electrode. That, as I said before, that glass at the bottom does have to be looked after. That does affect the way that the meter will work. Right, buying a pH meter. There's a huge range out there. You can go from the cheap Chinese pen types, which are ridiculously cheap now, up through different pocket types, and then right up to the sort of laboratory kit that one sometimes sees. I would probably say that a lot of it would be down to convenience. The benchtop kit's just too big, bulky. Some of the smaller pen type ones are just so much easier to use. I've gone for one of the pen types because it saves me having to use an awful lot of wart in order to take readings and so on. I'll show you the one I've got later on. I mean, again, costs comes into it. You could spend hundreds of pounds on a pH meter, or 10 or 15 pounds, and anything in between. The lifetime. Now, this is important because the probes themselves, the working bit of it, they do only have a limited lifetime. And this does again depend on how you use them and how you look after them, but most probes aren't going to last more than 2 years at maximum before they start misbehaving, and that's just because of the way they're made. Because there's exchange of ions between the sample that you're measuring and the inside of the electrode, they are going to eventually over time wear out, for want of a better expression. So this is important when you do buy a pH meter to, if you go and spend, which you can, £200 on just the probe for a meter, be aware that that's still not going to necessarily last any longer than a cheaper one. You can get pH meters where the probe's separate or replaceable. Sometimes, to be honest, though, the difference in price of the probe and the actual head of the unit is not a lot. One thing I've done is I have gone on eBay and other similar sorts of sites to look at secondhand. I've got a couple of secondhand pH meters where they have a separate probe, which you can buy. You can buy the probe separately, and one of them was a £300 meter. I replaced it with a £25 probe. I mean, the meter would cost me only £20 or something because it didn't have a probe. So second-hand ones on eBay are worth looking at. But otherwise, it's really up to you. I would say that the cheaper ones, you get what you pay for. It's quite easy to find out whether they work or not when you get to the calibration process, which I'll get to soonish. That's the one I've got. It's made by Hannah, who are a quite well-established firm. They tend to specialize in pH meters. That's the 2 points, as you can see there, because again, some of them might only read to 1 decimal point, which is fine, but it's nicer to have the 2 decimal points there. But that's one I bought for next to nothing off eBay. It didn't come with the probe, which you can see on the right-hand side there. But I got the probe. It's a reasonably good probe. I got that for not a lot off the internet. This one here has the settings on all of this. You manually correct it. I'll mention that in a minute when I talk about calibrating. And this one's designed to have a temperature probe. Either some of these some electrodes will have the temperature probe built into them. Otherwise, you will find pH meters that have got a separate temperature probe, and I'll go into that in a minute. It mentions slope here, which again, when I get to calibrating, I will mention a bit more. Right, calibration. That's sort of the kit I use to calibrate and read when I'm taking the readings. I've got a couple of measuring cylinders which I can put the buffer solution, which I'll mention in a minute, into something to put some distilled water in, nice little test tube rack, test tubes. I take my samples using a syringe, 10 ml syringe, because I just find them convenient, and they do tend to last if you wash them out pretty soonish after use. I just take a 10 ml sample into a test tube, and that's plenty for the pH probe to get into. Now, you can see the pH meter that I've got's got a very narrow probe, which I do like because that cuts down on having to have half a cup of sample. It means I can have basically 10 ml samples, and yeah, it's a nice little meter, that one. These meters do have a replaceable probe, but the probe's the majority of the price of the meter. And right, calibrating. The calibrating process is basically you're using buffers, as they're called. Now, what you have is you want a known pH above the pH at which you're trying to measure and a pH below. So basically, the buffers give you a means of calibrating the pH meter. So you can see on here, I'm actually using 6.86. I'll just show you a picture of the buffers. Right, the buffers here, they come in powder or liquid form. The liquid form you just have to use as is, and therefore I use the powders. These are the ones I get off the internet. They're pretty cheap. What you don't want to be keeping your buffer solution for long, so this method is it's a good way of doing it. You basically get this sachet, put it into 250 mils of distilled water or pure water, and then you've got 250 mils of buffer solution. The buffers are made at various different points. They tend to be 4.01, 7.01, 10.0. And the idea is that you want to set neutral point, which is why you'd use a 7 or a 6.86. Now, these ones happen to be sold as 6.86. They don't do a 7, which is fine because my pH meter enables me to dial in what the buffer solution is set at. some pH meters have auto calibration, and they will, you put it into the liquid, it can tell that it's 4 and therefore sets it to 4. You put it in a 7 buffer and it will set to that. But basically these buffers are so that you can calibrate between the range of what you're trying to measure. If you were measuring above pH 7, say between say 8 and 9 and so on, you'd use a 7 buffer and a 10 buffer. If you're measuring below, say where we are with beer, you'd use something something that would straddle that, so basically a 4 and a 7 or 4.01. And you can see that they say at 25 degrees centigrade. This is a very important point about measuring pH. It has to be done at 25 degrees centigrade. You take your sample wherever it is in the process and get it to 25 degrees and then read the pH. pH is very dependent on temperature. If you take a sample of your hot beer during the mash, say, and you've got your hot water during the mash, and you immediately try and take a pH reading, it'll be reading lower than it should do. And the other thing is it will damage your electrode by putting it into hot liquids. They're not, unless they're specifically designed for it, pH electrodes, again, are designed to work at 25 degrees. So your buffer solutions, when you're using those in the calibration process, then you want to be doing this at 25 degrees ideally as well.
Just to say, Jerome, those powder buffers that you can buy cheap off of eBay and other places, they are good. I tested them against some much more expensive Hannah buffers, and they were dead on.
Yeah,
if you
right.
use them as described, like with 250 ml of distilled water, mix them up, and then they last a while, but I wouldn't keep them for months and months before because it will start to drift after a while. But yeah, mixing them up, they were absolutely dead on.
Yeah, you're right. Yeah, they are good. And you're right again about how long to keep them for. I can't remember. I think it's the pH 4 lasts a lot longer, being nice and down there. The 7 pH buffer being at a nice pH for growing things does tend to go off sooner. You end up with a sort of growth starting in it if you do leave it too long. And of course, therefore, you've got a change in the pH. Having said that, buffers are in the name of them are very good at resisting the change in pH from something.
Jerome, do we know why the numbers aren't rounded up to 9, 7, and 4? What's the deal with 9.18, for example? Do we know?
Oh, you see, where they have this. Well, I'm not quite sure why it's 4.01 that is picked,
Yeah,
for
that.
instance. Yeah, I don't know actually. No, because I mean, you can get buffers which are, I can't remember the name of them, which are the very special scientific ones, which you can get within 2 different points. So you get whole sets of buffers and so on. If you're trying to measure below 4 or above 10, then you'd have to get special buffers in. But then I think when you get too much lower than sort of 3 or 2 as a pH, you're going to start damaging your equipment anyway.
I mean, I guess it doesn't matter what the number is, as long as your gear calibrates to the number in the solution, doesn't actually matter. I just wondered why it isn't rounded up.
I think the answer is it's like they've got a chemical there that is a good buffer that dehydrates well, and it's a pure sort of chemical, so you're not mixing it with other things. And when mixed with distilled water, that's the pH it gets to, and it's probably the closest thing you can get to those numbers.
I mean, they will, as you can see on the packet of buffers, they've got information about what the pH would be at different temperatures there. So if you are actually using your buffer, you should be aware of that if the temperature, and you could put that into, with some devices, you can use that information to change, which will probably bring me along to yeah, I've mentioned temperature as being important. The point calibration, you can do 3-point calibration. If you were really being fussy with your equipment, you could do 4, 7, and 10. And a 3-point calibration will be better. And what you end up there is a slope, which on a graph would be a straight line going through the center point, and the sharpness of the slope will tell you something about the condition of your electrode. How long it takes to get a reading stable will show you that you might have problems with your electrode. So it should only take 1 or 2 minutes to stabilize or whatever. If whilst during calibration it's drifting all over the place, not being stable, then you have a problem. This is where I say rinsing in between doing anything is important, to make sure that there's nothing residual on the electrode. Yeah, again, where you will find the phrase automatic temperature correction written on pH meters. A lot of people wrongly assume that the automatic temperature correction allows you to put the pH meter into anything and it will assume that it's 25. The automatic temperature correction is actually when you're using the buffers. So if you've got your 6.86 or your 4 buffer or whatever, if it's in that buffer, the device will compensate for slight changes in temperature to accommodate the calibration, but that's at the only time when the automatic temperature compensation comes in. So that's just for the calibration process. It saves you having to be able to precisely put in the temperature of your buffer solutions when you're calibrating.
So, Jerome, I think I've managed to destroy a few pH meters over the years. Most of that has been in relation to sour beer. So I would calibrate to the 4 and 7, but then measure pH in the 3, 3.5. But they never seem to last. So what you're telling me makes a lot of sense, is that if you're measuring outside of what you calibrated to, then you could destroy, well, the electrode just doesn't last as long. Is that what has happened?
I'm not sure it would destroy it. I mean, I think you probably have to go a bit lower. I mean, you have to probably go to, it's just that if your buffer is 4, measuring anything below 4 isn't going to be very accurate. I mean, it should be, because what you're doing is you're producing a calibration line. So you
Yeah,
should
it's extrapolating, yeah.
you should be able to extrapolate up above and below that. But
I found it would sort of converge quite quickly on the first like 5 to 10 reads, and then after that it would I just throw it away, because it would just be all over the show and just keep ticking over even after quite a while. Okay, yeah, I think that's what's happened. It's just not as reliable for as long if you're measuring outside. But
yeah.
okay.
Right.
And Jerome,
Yeah,
sorry,
yes.
I've got one of the Voltcraft meters, which has a separate probe, and they're quite good because the separate probes are, like you mentioned, are dirt cheap off AliExpress or eBay or whatever. And I can't find anything actually in the manual about temperature. And for some reason, I've always cooled the water 20. So are you saying that the buffers clearly will have a temperature in order to read?
Do you mean, well, I'm just saying that
6.8, if they claim ATC.
what I'm claiming is that we're saying is that ideally you have your buffers at 25 degrees C when you're calibrating. If they're not exactly at 25 degrees C, the automatic temperature compensation does allow that's where that would kick in. So when I'm calibrating, I don't calibrate. If I've got my my buffers, I usually keep them in the fridge. I'll take them out an hour or two before. Well, in fact, what I do is I'll take the buffer out and just pour a certain amount into a measuring cylinder, which I'll just chuck away when I finished with it, and that would mean that it'll be up at room temperature.
And that's close enough to 25.
Yeah, because the automatic temperature compensation that's what that is for, is just to compensate for any difference from the. I mean, you're not looking at a huge. I can't remember. You're not looking at a huge difference between, say, 20 and 25 or 25 and 30. But if you're up in the 60s degrees, that will make a considerable difference.
Yeah.
Right.
And
Just
the storage solution, that's quite a high pH at the storage solution in order to keep the probe in good, in good.
Yeah, I'll
nick.
mention that in a minute. I'll
Okay.
just do the
Sorry.
calibration, then I'm going to do the, So calibration, basically, be aware of the temperature of the sample, get it to 25. You could use a sort of water bath with cold ice, whatever, if you're in a hurry. I would say that a lot of these things, when you're measuring the pH, probably by the time you measure the pH, it's probably too late to do anything there and then about whatever the situation is. So I tend to take my readings after, an hour or so after I finish brewing, by which time the sample's got down to temperature anyway. Rinse the probe, then set for pH 7. You set for the midpoint first. So always set for 7 first, and then do your either your upper or lower point, or both if you're doing a triple point calibration. Rinse again. Yeah, you set to your upper and lower point, rinse again, and then you're ready to use. When you're using the electrode, be careful not to let it hit the bottom of the sample jar if you've got a bare electrode. The probe I've got's got a handy sort of plastic protector on it, but do be aware that that glass electrode is thin and can get quite easily damaged. So by all means, give the sample a slight stir. You want to make sure that there aren't any bubbles or anything stuck on the probe when you're taking your reading. And it's good practice just to give the sample a slight stir, but don't get too carried away with using the probe as a stirring rod. You can see on this that this is top of my pH meter. The screws are looking rather rusty, actually, but you should be able to see there that there's an adjustment screw for setting pH, and on one side you've got pH 7, and on the other side you've got the 4 and 10. So basically you'd be using a small screwdriver. On this one, you'd use a small screwdriver to adjust the reading so that it was reading correct on the display. Buffers, I've done the just quick one on the buffers. Yeah, don't keep them for too long. They're cheap enough that you can certainly, I'd say 6 months maximum. Because otherwise, these are the things that are going to, they're your calibration tool. So if these are off, then everything else is off. It's worth, if you do mix up some more and you've got some left at the end, of just doing a comparison. I mean, I do that occasionally just to check that a 6-month-old buffer hasn't really gone off, and they don't. 6 months is fine, because I'll check the old batch against the new batch.
It's only like 60p a sachet.
Yeah, it's not worth. Yeah, if you're going to do this, then you might as well do it. Yeah, the care and maintenance. Rinse all the time in between anything. Get anything off that electrode as soon as possible. Don't let the electrode sit in the sample for longer than necessary. It's not good for this. It's basically because the process is constantly occurring, this ion exchange, you are going to, with want of better expression, wear out the probe, and it'll be made worse by certain chemicals and so on. You'll find that. Go on.
Just on the ion exchange, does that work kind of like how a battery works?
It is. It is
So
basically.
basically, it's a one-way street,
It
and
is
it will
exactly.
eventually just run out.
It is basically, it's a galvanic process. Yes, you're basically the pH meter is a cell, basically. It's a battery you're sort of putting in you're creating a battery between your sample solution and your reference solution, and that battery is creating an electrical potential, which then you're amplifying, and then you've got your reading. So basically, pH 0, there's no voltage, if you see what I mean. So sorry, pH 7 at neutral, there is no voltage. So you're basically, your slope would be either sides of a 0 point, so to speak. You're only measuring the hydrogen ions. So in other words, you're measuring what the acid part of the solution, if you sort of mean. That's what you're actually measuring. You're not measuring the hydroxide, the other side. You're measuring the hydrogen ion concentration.
So when you're above 7, are you running the battery down, as it were?
I think I think because you're at neutral point. Yeah. Don't quote me on that.
No, I didn't know that's how it works. That's interesting. Thanks.
The cleaning. You can basically, a good rinse is usually all you need. Wiping is, be careful of wiping the electrode. By all means, wipe off excess off the body, the protective body, and so on. But try not to wipe the electrode itself, because you don't want to scratch the sensitive coating on it. A dab with a lint-free tissue or whatever is fine, just to get liquid off it, but literally just dab it on, take it off. Because there's another problem, which is if you rub the tip of the electrode with anything that could produce any type of static, then that's going to set your pH meter in a bit of a peculiar place, because basically you're charging it up, which is going to then throw all the readings. So you don't want to be rubbing it particularly, but mainly just to stop the electrode tip itself, the membrane, getting damaged. If you get a lot of protein building up on the probe, and you tell dirt on the probe, well, basically it'll show itself as the probe values drifting, not settling, or taking a lot longer to get somewhere. So two indicators of a problem with your electrode will be it taking a long time to stabilize to a reading, or that it drifts. It just never, it just basically, even when it gets to something like a reading, it'll just keep moving around. Storage, you want to be storing them in, well, basically the storage solution isn't that dissimilar to the pH 4 buffer solution. It's because pH 4 buffer solution is potassium chloride, which is actually the same stuff that you would use to store and if need be, reinvigorate your electrode. I buy this the stuff on the right from, again, the internet. This is stuff used by people who grow plants indoors. So, or people who have fish in tanks. It's dirt cheap buying it that way. It's much better than buying it from other places. And basically, it's potassium chloride, which is actually one of the constituents of the electrode itself. One of the chemicals inside the reference electrode is basically potassium chloride. So by soaking the tip in something the same and at the same pH and molarity, the strength, so to speak, then that protects the probe. Always keep the probe stored in the solution. My pH meter has a rubber cap. I squirt in some clean storage solution, push the cap on until the solution is expelled to excess, and then I know that the thing's nice and wet. You must keep the electrode soaked wet all the time. Drying out is probably the main reason why they fail, because that tip has to be kept moist for it to work properly. You can revive the tips if it does dry out. Stick it in some storage solution for a few hours, rinse it off and see what happens. If you get protein build up on the tips, then a bit of ethanol, bit of alcohol will get that off. If they do get dirty, you can, with other ways, you can use 1 molar 0.1 molar hydrochloric or sodium hydroxide, but I wouldn't bother with any of that really. I mean, you're most likely to get a bit of protein build up, which again, a mild detergent, a soak in a mild detergent isn't going to do it any harm. Then rinse and into storage solution. Going back to the calibration, I calibrate every time I do a set of readings. It doesn't take more than 5 minutes. So I would calibrate every time. Obviously a brand new pH meter you would calibrate, or if you get a new probe, then you'd need to calibrate it. Again, probe life, they don't last forever. So just be prepared that if someone's got a 5-year-old pH meter and don't understand why it's not working, it's probably just had it. So it is, again, be careful buying something really expensive. The breweries will change theirs every 6 months, and they might be spending £200 on a Hannah probe. Right. I think that's about it. I mean, I can talk. I'll just show a little bit of the process, I suppose, if you bear with me 1 minute. Right. This is my setup, sort of setup I'd use. Got the pH meter here, and you can see it's just see the glass electrode peeking out the bottom there, that sort of whitish object. Right, so what I'd normally do is I'd be rinsing it in a bit of distilled water. I then handily flick off any liquid. You possibly give it a little bit of a wipe with a cloth if need be, but then it would go into the buffer solution to be calibrated. Now, because I only set this up earlier on, I'll do it correctly and put it in the neutral buffer first. So this would be, just get that a little bit closer. So that's stable. That's in the 6.86 buffer solution. I haven't used this particular meter for a while, so there it goes. It's climbing up. Now, the faster this occurs, then the better your meter is. I mean, this, I'd say that this one's getting a little bit old. That's because it's taking what, 30 or so seconds, which isn't too bad. Where are we? It's starting to stabilize now. Now, as you can see, that is higher than the 6.86. I'll just let that. Right. Assuming that's pretty well stopped there. Well, once that's stabilized, I would then take the pH meter. I would take the pH meter. I'm just checking to see which one's which. Yeah. And I would then adjust with the screwdriver this until it was reading 6.86, which is what that buffer's meant to be. It's quite sensitive, that. So that would be setting 6.86. Then it would be rinsed again. And then we'll go in the pH 4 buffer. Oh, closer. And basically, when that gets to 4, which it is doing, and if it's above or below 4.01, then I would again adjust that with pretty well. So you see, it's either here again. It's dipping down a little bit below what it's meant to be, but I can then adjust this. Whoops, wrong one. I'm not looking properly. I'll just check this one. And then again, I would adjust this until it's 4. So I've then now got it at 4 and 7. And then when I put it into my sample, whatever it reads, I mean, I don't even know what this is in here, but it's beer of some description. And when that stabilizes, that would be me taking the pH reading. Because I'm being fussy at the moment, I tend to take sort of pH readings, well, just because I'm interested, at different points through the mash. And that's interesting. I tend to take a reading at, because I've been doing 75-minute mashes recently, I'll take a reading at 60 minutes, 50 minutes, 25, and 0. And what I find is that the pH is, after 15 minutes, is lower than the finished pH. It takes probably 30 to 45 minutes to for me to see the pH stabilize, which is interesting because it's a process that's, as the mash process is occurring, the substances that are being released into the mash are changing the pH, and different things happen at different times. So there we go. It's pretty well stable at around this reading around 5. Yeah, that's not too bad. As I say, this probe isn't my number one. It is getting a little bit old because it's it is taking a while to take that. I'd say anything over 3 minutes, and I'd start to get a bit like, how reliable is this? But then if I was worried about the reliability of it, say if it was drifting and taking a while, I could check that quite quickly by sticking it back in the buffers and just checking to see what's going on with those. If it doesn't, if by putting back in the 4 or the 7 buffer it was wildly different, then I know that there'd be something wrong with the probe. And the thermometer, of course, is vital for checking the temperature, which is what I didn't do this evening. So the temperatures of the buffers might be a bit odd anyway. I think that's about it, unless anyone's got any questions to ask.
So in terms of the pH values during the process, is there anything you particularly aim for? Or is it a case of you just sort of measure it and adjust it next time?
I measure and adjust. I can't see how you could know sort of what you were doing if you were doing it on the fly, if you see what I mean. But yeah, my actual aiming, I aim for 5.2 in the mash as a final. Well, 5.25 as a final is where I'm going for. But that's because I'm doing a lot of hoppy beers, and I find that, and also have read that with a lot of hops in your beer in the fermenter, you are raising the pH noticeably. So I go for a lowish pH.
That was probably going to be my question, and I see Serge asked it in the chat group, is earlier on you were showing IPAs and American Pale Ales at a pH of around 5 to 5.5 or something. Is that right?
Well,
I guess the next question is what is a good pH for an IPA? I've heard a lot of people say it really helps if it's a higher pH, or is it just because from a taste perspective, or is it just because the hops increases the pH and that's where people land? But what do you really want from an IPA? Do you want it at a lower pH or a higher pH, or what's the sort of taste versus science? And
well, from what I gather, that a lower pH will produce a crisper, less muddied sort of taste, apparently. I mean, there is that question as to whether it's the extra hops, because the extra hops do bump up the pH noticeably. So a heavy dry hop charge will push the pH up. Scott Janisch did an interesting article about the effect of hoppy beers on the pH. And so it's a known, not quite well understood what's going on, but it's certainly a known phenomena, to the point where some breweries who are producing American style beers are adding acid to their fermenter for fermentation because they're not getting it to drop. It's not dropping through the process enough. I mean, the pH will drop all the way through the process. You know, your boiling will drop it, and obviously your fermentation will as well. But there are brewers who are putting in phosphoric acid, say, into the fermenter just to push the pH lower, possibly to counteract the effect of a high hop charge.
Well, on that, absolutely. Like all the evidence shows that the dry hops massively increase pH. So if we take the final pH of beer, for simplicity's sake, as a function of the yeast itself, because what you get with an ale yeast, let's say an English ale yeast, your English bitter, can quite easily end up around 3.7, 3.8 pH at the end of fermentation, whereas your standard lager yeast would quite easily end up around at 4.2. For those with pH meters, and I really stress this, do not kids do not repeat this at home. You can play around with a small sample of beer. It can be a commercial one, especially let's say a lager, which is maybe like a Vienna lager or something. And if you just increase the pH slightly by dropping some dry hop in there, or lower the pH with acid, what you will notice is that beers, especially the hoppy beers around 4.5, or at least pushing that 4.5, have a much more malty appearance. You take a NEPA, for example, but if you drop it with acid to closer to 4.2, you end up with a more sharper, bitter perceived beer, and you lose that maltiness. At the same time, higher pH around 4.5 makes the flavor and your beer not as stable as it is at the lower pH. So it really kind of depends what you're trying to achieve and what you're doing with your beer. But it's very interesting to see how different beers behave. And I have successfully, at least to my palate, adjusted lagers down to pH and gotten better flavor again to my palate. And this was done post fermentation. I have also screwed up once, and I had to dump 30 liters of a Helles because I just eyeballed the acid. And so and whenever you're doing that, please do not drink your new concoction before measuring the pH beforehand and giving it a good stir, because it is possible that you will overadd the acid and you will hurt yourself. I really can't stress this enough.
I've done the same thing actually with dropping lactic acid in flabby lagers to crisp it up, and measuring it properly as well. And it worked for me as well. It's pretty good, and I thought it was noticeable. I kind of lined up a few 5, 4.6, 4.2 pH lagers and out of the same batch, and you could tell the difference. It's got that nice lemony twang, but it's crisped it up a bit as well, took that flabbiness out of it.
So how do you adjust it, Steve? And like if you're using lactic acid, how much? How do you calculate how much?
You can only figure it out by trial and error, right, Steve? Because your buffering capacity of a beer depends on each beer. It will be very individual.
Exactly. The buffer can change. Yeah, exactly.
Yeah, if you brew the same recipe 15 times, you know exactly what it's going to be, right? So you could probably even add additional acid before fermentation is finished, so that it's properly mixed in. If you're dosing afterwards, the mixing can be a problem, because you take a small sample, you add a little bit of an acid using a very precise pipette. Yeah, then you dose the same amount of acid, multiplied by the amount of the beer you have in the fermenter. I don't know, give it a day or two. Sometimes you're like, I've just overdone it, or I've just underdone it, but you don't know how well it's mixed. So
I've had exactly the same process and experience, Serge, as well. I think exactly. I started with like 1 drop of lactic in a pint of the lager or something like that, mixed it up, measured it again, blah, 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 blah. And then tried to multiply it up per keg. And sometimes I got it right and sometimes I didn't.
Definitely better to underdo it than overdo it.
Yeah, for
you can't go back.
Yeah, I had to dump 30 liters of a Helles. So.
And lactic acid does have a flavor, so it's do pick it up.
Yeah, but I would say, look at, if you're trying to improve your beers, this should not be in your top 10 to improve your beers. This is more for like geeky fun, even.
It's definitely what the Americans call a Hail Mary of try and salvage a beer that's just not there.
Well, I mean, it's a little bit like the whole thing about measuring pH anyway. I mean, a lot of good brewing software will get you there without it anyway. I mean, if you follow good instructions from people, then you should be able to get there without. Having said that, it is remarkable what will affect the pH. I mean, different malts from different maltsters, because they'll have different buffering capacities because of the amount of the proteins and so on, distribution. The fact that dark malts will push the pH down, light malts tend to do the opposite. So very pale malts can have less of an effect of pushing the pH down. I mean, I'm reading that those say to the point where if you're using darker malts, you might want to leave them out of the mash. Well, leave them till later in the mash so that they don't affect the pH too much, because you're not after too much out of them, are you? It's not like you're after a lot of conversion.
Well, with particularly with London area water, because it's so high in alkalinity, when I was first dealing with pH, I was struggling to get pH down. Yeah, that was it was fine for dark beers, but for pale beers, yeah, I just wasn't getting it down. But yeah, dark beers with lots of roasted malt is fine.
Yeah, because of course they're pushing the pH down. Yeah, yeah.
Make an imperial stout, and I don't just use the tap water as it is. But yeah, if you're making a pale ale or something like that, then RO is much better. And I also found that the software that was predicting pH was just not very good at the high level of alkalinity. I think they generally got better, but the fact that you can use brew and water and you can use something else and they give you fairly different predictions gives you an indicator of how difficult this is.
Oh yeah, yeah. The buffering capacities of different chemicals involved in the process can change things massively.
And ultimately, for me, the pH itself doesn't really do anything. It's just a measure.
It's the activity. Yeah, I mean, if you were measuring pH properly, you are sort of measuring the activity, but you're not measuring the actual acidity, so to speak. You need to titrate to do that. I've rereading that people who do a lot of sour beer, producing a lot of sour beers, use titration as their method of working out the actual acidity rather than the active acidity or whatever, or the active component, the active pH.
So I just find it as I mean, I don't actually measure it all that often these days unless there's a particular beer style I'm trying to really nail, and then I need every data point I can get to say, right, well, this pH was a bit high here, so maybe I'm going to lower it down, or been messing around with Weiss beer recently, and the yeast for that produce quite a lot of acid, some of them. So I started like raising the pH in them at the beginning to try and offset that a bit. But it's just a data point. It doesn't actually it causes there's all sorts of other things that go on around it, but the pH itself is
I mean, basically, the brewing process is it's in the right pH place because that's what it is. I mean, a lot of it will sort itself out. Your protein break will work at a certain, well, it is a pH that's available within the brewing process. You haven't got to go anywhere because that's what brewing does. So most of the time you're within the ballpark anyway. It's probably of more interest to say if you change a particular aspect of your brewing, then and that will affect the pH, then it might be worth looking at. If you're changing your grains massively, or if you were looking for a problem that you didn't know why it was occurring, you might then think, oh, well, I'll just check and see whether using this malt has changed the pH, or, I mean, even crushing differently is going to change the pH because you'll be changing the amount of available extract slightly or whatever.
Yeah, I think for me it was just of limited value when I was brewing a different beer every time. It's when I started coming back to beers and rebrewing same sort of thing. That's when it really started to matter.
Yeah. I think that's only, I mean, otherwise it's a point of interest. But yeah, if you're after consistency, then it's worth looking at. And obviously those that are, say, making sour beers, being able to look at the pH or whatever is useful to know where you are in that process, because there's an element of control that you want to have there on your sour when you stop it souring or whatever.
I found by the time you know something's wrong, it's too late. Just like you say.
It's next time. It's always next time.
Yeah.
Well, you see, it's interesting that when they talk about adjusting the mash pH, for instance, well, if you're 15 minutes in and you take a reading, well, that's going to be 5 minutes later before that. Most of the conversion's done. So then you think, well, what happens now if I add acid now in the process? That's not the same as it being there at the beginning, because it's a different, you've got different chemicals in different quantities by the time you then add your acid. So if you add acid 20 minutes into the mash, you don't really know what it's going to be doing.
No, you're right, but I think here what's important is, sure, if you've hit your mash at, let's say, 5.5 and you want it at 5.3, okay, perhaps it's maybe it's a bit late, won't do much. But if you measure your mash pH at something like 6 and you're nowhere near the range, then possibly your conversion is slowed down and actually hasn't mostly occurred. So then bring it down into your 5.2 to 5.6 makes sense. So
Oh yeah, yeah. I mean,
you kind of have to be smart with what you're doing, really.
yeah, yeah. And I mean, it's not a never say never, whatever. But I'm just saying you wouldn't really know what if you were trying to quantify that would be tricky. You obviously if you need to change your pH, then bang in some acid. You get it. Yeah, get it. It's a drinkable beer.
No, definitely. But also what I've noticed, and so far it's 2 batches. I can't comment on, this is an anecdotal evidence so far. But what I started measuring, just because I feel like I have, a good grasp of many other aspects, I've started measuring my pH, not just in the mash, but also pre-boil, post-boil, and at the end of fermentation. And what I have seemed to have noticed is that I get a much better hot break in the wort when it just starts boiling and 5-10 minutes in, when my pH post mash is around 5.2 or even lower. I haven't looked up at literature yet in that regard, but it seems to ring a bell that I've heard something around the pH effects in your boil, not just your mashing enzymatic activity. And then post-boil pH should be around 5 to 5.2, if I remember right.
Yeah.
I feel like it was in Steve's presentation from a couple of years ago. And then your yeast has a better head start at fermentation rather than trying to bring the pH down to the level where it's more comfortable fermenting.
I'm actually literally looking for a pH chart that Murphy and Sons had, which is exactly what you're describing, Serge, that pH throughout the brewing process. So you're mashing the beginning of the boil, at the end of the boil, beginning of the fermenter, end of the fermenter, finished beer. There's a kind of a specific range of pH to be aiming at all the way through the process. I can't find it, and I might have used it in my presentation. I've got a printout somewhere, but I'm not going to go searching through all the piles of paper. It's somewhere on the computer or the internet, but I cannot find it. I've been looking for it for about 10 minutes. If I find it, I'll post it in the in Slack or something. But it was a really useful chart, and I used to pin it up like on brew day to make sure I was getting to. So for example, mash was between 5.3 and 5.6. At the end of the mash, you'd be 0.1 pH lower. So the end of the mash should be aiming at something like 5.1. And then the end of the end of fermentation was something like 4.4, something like that. And then your finished beer was something around that 4.2, depending on the style. Obviously, if it's sour beer, then you're 3.5 or something like that. But generally looking at like 4.2, 4.4, something below 4.6 to make it food safe, to keep it below where botulism will basically be killed by the pH, but also you've got the alcohol will kill it as well and everything else. So making it shelf stable and food safe, I think below 4.6 was the thing. The original, I don't know if they still do it, but the Brewdog PDF of all the recipes had a pH of their finished beers, I think, on every one, and it was something like 4.4, something like that. It was basically the same for everything.
That's I thought that it's interesting that there was a slide earlier that had the pH of finished beers, and some of them were well above 4.6. But then
Yeah,
there's also the idea that 4.6 makes it food safe, and I wonder if there isn't, as you just suggested, a combination of alcohol and pH that it's not just a one-dimensional safe threshold. And it would be interesting to know, because when there's a lot of interest in brewing lower alcohol beers, and reluctance to judge them if you don't know that they're food safe. If there is a simple way for us to measure that they are food safe, for example, just by measuring pH, or by making a threshold for we will judge things with pH below, a certain level in order to, health and safety kind of thing.
I made a
I don't know,
few.
just a thought.
No, I made a few 0.5 pale ales, and I was really particular to make sure that the pH was well below 4.6, so that there wasn't any chance of anything happening, as it were.
Yeah, I must admit I'm surprised by those figures that even that I produced. I must admit when I was reading them, I did look up on two different sources for those high IPA pHs, because I was surprised myself to think, well, hold on, if the FDA, the American Food and Drug Administration, have got a 4.6 on packaged beer, what are you going to do if it's above it? I mean, a lot of the testing at packaging is it's a pH is purely to get it so that it's considered food safe. So I must admit I was a bit surprised by those high figures.
Yeah, I mean, it's also interesting that we in the dry hopping, throwing the hops into a barrel days, that they were introduced, or maybe this is apocryphal, but they were introduced to help preserve the beer, and yet they raised the pH. So I'm imagining there's some subtlety there that probably isn't captured by FDA rules.
I remember reading this article about they spoke to the brewer from Weihenstephan and asked him what's the most important part of your process, and he said the boiling, and this everyone was surprised, and he said the pH of your boil is the most important part of making Weissbier, which was I found interesting. He says it has to be at 5.2 because that's when you sort of you get the right balance between the amount of proteins coagulated versus the good starting point where you want your yeast to start at. It also increases stability of the beer. And I think as homebrewers, we don't really worry about this. But for larger brewers, they do. You want maximum protein coagulation for the stability of the flavor of the beer over time. Maybe for stronger beers, that's probably even more important. And he said most of the time people boil at a slightly higher pH, but target 5.2 is what he was saying. I think that might be different to different beers, but I remember boiling at a higher, because sometimes it boils and it just looks like water boiling. And then I just added a little bit of acid, and it was almost 5 minutes later, you could see the proteins just sort of floating around in it, and it made a massive difference. I don't have a like-for-like comparison, but it's really interesting how for some brewers it's extremely important.
Yeah, Jack, and that's exactly what I found anecdotally in the two batches. Why I was like, okay, let me see, if I can just play around with this aspect of brewing, because you need to do new things, otherwise Bruins are same old all the time. And this is exactly what I found. You can see the clumps.
Yeah. Yeah, if you see the clumps, then you sort of know you're boiling well. It's not something we ever think
I
of.
guess so, but it looks bloody disgusting. It's like as if you're boiling like some mushroom sick in your wort as well, yeah. And then, at the end of the boil, actually, there's a very interesting blooming effect. They clump together, like as if you're making cheese curds.
And then they settle out at
And
the
then
end
they
of the boil.
settle out, yeah.
Yeah. Hmm.
With regards to the probes, they've got a limited life, but do they have a limited life from the moment you start using them or from when they're sat on a shelf?
I reckon it's from the day they're sat on the shelf, to be honest. Because let me think. Well, I don't know. I imagine that if they're kept in the correct storage solution and both the tip, because there is another little membrane that gets involved in it, which is between the reference cell, which I didn't bother to mention because it's more complicated than I quite understand. But they're basically, I'd have thought that if it's been in its storage solution, it should be all right because it's going to be in a neutral position. It's not going to be losing or gaining any ions because the solution inside is the same as the solution outside. So it's balanced between the membrane. So I imagine, unless it's dried out, then it's probably all right. But I think once you start using it, you are going to be, every time you use it, you're going to be affecting the concentration of ions in the reference and the outside. I mean, it's just one of those things. I mean, I know that these companies try and get you to buy new whatever all the time, but it is significant that a lot of breweries, I was reading when I was doing a bit of research and on the best one, which I think comes up, the people swear by the either the Hannahs or the Genways, another another one, and I think they're the probes on the most popular one that they were talking about seemed to be about 100 quid, but they were talking about every 6 months or whatever. I'd probably say that's down to possibly the people using them not necessarily being as careful as, if it's in a brewery, depending on who's allowed to do the lab work, if it's just given her out amongst anyone to mess around with. But
They're just jamming it straight in the mash.
yeah, exactly. They're in a hurry. They're not going to let that cool down. Oh, they're going to rinse it properly. Yeah, yeah.
I mean,
I'd certainly say, I reckon that unless you see it taking ages to say, I mean, you can tell when a probe's gone wrong because it's not reliable. You should be able to take it out of a pH 4 reference buffer solution, rinse it, stick it in a 7 and it should read 7, stick it back in the 4 and within a short while it should read 4 and so on. If it takes ages and ages to do it, or it doesn't go back to where it should be, in other words, it's drifted, then, that's probably a problem.
Yeah, I had an Ampera one, which after a while I thought, what the hell's going on with this? And then I got the Voltcraft one, which is obviously a lot better. But then I'm thinking, as Ian mentioned about buying probes on AliExpress, and then I'm thinking, perhaps I get a few of those in stock. And then I'm thinking, hold on, what if I get a few of those and then
No, don't keep them in stock. I mean, I genuinely, if you know what you're looking for, the the eBay route can be great. I mean, I have about 4 pH meters, and one of them is it's a really nice Hannah, and it's a top of the range. It must have been, I think it was 600 quid new. Now 200 quid of that is the probe, but the rest of the kit, it was 30 quid or something on, or 30 or 40 quid on eBay, because the probe was knackered. Well, the only thing about the probe is it has a different connector. So I'm just saying, if you do buy a second-hand one, do check the connectors, because they can be a bit crafty. Some of these companies are putting a peculiar DIN plug or whatever on the as the setup, and the only thing that stops you being able to use a generic pH probe is the wretched connector. So I would check the connector. I mean, they tend to be what they call BNC connectors, like the sort of phono type, where you, and it's got a tend to have a little locking ring or whatever and 2 prongs. Those are the standard, but there are some that have a fancy 5-pin, because they've got a built-in temperature probe. They're running 5 wires or 4 wires into the unit, and so they can have annoyingly non-compatible. I mean, it's nothing that you can't, nothing to stop you buying a cheap electrode, cutting the wire off, putting on a different connector if you're up to that sort of thing. But it is something to be aware of. If you buy a second-hand one, just check that the connector, if they've got photographs of it, is something that's compatible. It's usually a BNC connector. But as I say, some of those eBay ones are absolute bargains, because the person sells the whole device because the probe's gone, without realizing that you could just buy a new probe for them.
Yeah, I mean, I've got the Voltcraft one as well, and the Voltcraft probes, actually, when looked after properly, have lasted quite a long time, like years.
Yeah, I reckon you could get at least 2 years out of it without worrying. I mean, as I say, it's only when they start playing up that then you think, and you can tell because of the idea of drift and so on. I mean, if you could be bothered to actually plot the straight line and use the Nyquist equation or whatever it's called, which one of the meters I've got, you actually can do that. You can actually, if you can be bothered to, it gives you a value and you can actually plot the slope, and it'll show you whether the thing's iffy or not. But usually they just start drifting and misbehaving. You take them out of a sample and you think, well, that's a bit odd. That should only be a couple of points difference, and it's suddenly reading 2 units difference. Then
yeah, I mean, the cheap replacement probe I got from sort of eBay that was about a £5 or something was it wandered around a little bit.
But that's a £5 though.
Yeah.
I mean,
So
if you spent 15 quid.
Yeah, so there's definitely better and worse one. And then I went back to I bought another Voltcraft one, which cost a whole load more, but and then that was absolutely fine and lasts for another few years, and I've still got on mine, but I'll have to check to see whether it's usable. To be honest, I haven't used it in a while.
Yeah. No, it is the advantage of I mean, you either don't use them a lot, in which case that is the trouble, I suppose, with the little pen ones, the pocket ones, is that if you don't use them a lot and you don't look after it, it then becomes a useless item, because once the electrode's gone, the whole item's useless because they're all built into one. Whereas the separate probe, I'd say, well, the electronics of a pH meter will last forever. I mean, they're not, sort of forever. They're as reliable as anything. They're just a sort of bit of electronics to amplify the signal. So a separate probe, as I say, is useful in that respect.
Have you covered it? You're talking about the cheapy ones. Obviously, I started off getting one of those, and I think I've got a slightly more or slightly less cheapy one, but they've just got the little caps on them. So I haven't read. One of the bits that you mentioned is about keeping them, in wet solution all the time. And so when you get the little ones, they just got the cap. So I've just used them, dried them, or rinse them, and then stuck the cap back on. So clearly that's not going to keep it going at all, is it?
No. What you have to do is You mean one of those like a rubber cap like that? I
No,
mean,
the
basically.
square yellow ones, you know, the
Oh right, no. I mean, basically, that's the cap that comes on this one, and
yeah,
it's not a rubber cap. What I would do with this is I'd fill that with the storage solution, and then I'd put the cap in until the storage solution is then pushed out, which you can sort of see possibly. You know, it's running out now.
yeah.
That I know is now soaking wet inside, but it's not the best way of storing. I mean, ideally that would be left in a something, you know, like a test tube or whatever. But then you've got the worry of it getting knocked over. But that cap really, the electrode must be kept wet all the time.
Yeah, I'll have a look at it because I think I don't.
And you can restore it if it hasn't necessarily killed it. What you would do is you'd leave it in the storage solution to soak for 4 or 5 hours or overnight or something, and that quite often will revive them.
Forget about it, and then test it again, and if it's
Yeah,
okay.
and if it's not, it's not. But you can revive them. I mean, I have read where people have said, "Oh, I've done, you know, this is in chemical laboratories," where someone said, "Oh, I stuck it in, I don't know, sort of whatever it was, caustic, and have I knackered it?" And they'll say, "Well, no, put it back in potassium chloride solution, what the reference cell is made of, stick it in that, and it might just clear itself."
Is it important to get the storage solution, or you mentioned that the 4 was close to it? Is it worth?
You can stick it in 4 and you'd be perfectly all right, because 4 is basically it's pretty well the same strength and pH of the. But having said that, that stuff that I showed the bottle of earlier on, the hydroponic or whatever it was, I mean, it was like a couple of quid for a bottle like that. It's going to last me my lifetime. I mean, I'm using about 3 ml a time. So I've got 100 ml bottle. So I'd just go on eBay and look up sort of pH storage solution, because as I say, a lot of people, anyone who does hydroponics or fish, you know, saltwater, well, you know, tank fish sort of stuff they'll have a pH meter as well. In fact, they're far fussier about their pH levels. Some of these reef enthusiasts, you know, are trying to
Yeah, got to get their warm tropical fish.
Yeah, and they're talking about being very, very. I mean, the seawater is it's in quite a narrow range. You're certainly within 0.1 of a pH here and there. Yeah, keep the thing moist all the time. Yeah.
Yeah, but I think one of them's definitely I've had for some time, so I'm sure that one's knackered, because not only is it old, it's also not been kept at all. So I might discharge that one. I'll see if it works.
Well, give it a go.
Yeah.
I mean, it's like all of these things. It's worth comparing one with another. I mean, it's like if you've got a hydrometer and you have another hydrometer, it's not wrong to think, oh, I wonder whether these things actually read the same.
Yeah.
You know, and they might read the same in water, but they might not read the same when, I say, a hydrometer at 1060 or something. So
yeah, I'll have a test.
I found that printout, Jerome. I've dropped it in the meetings. Took a photo and dropped it in the meetings in Slack. Oh, God.
Oh, right.
Why is my face in focus but the stupid printout isn't? Anyway, it's in
it's
much preferable the other way around, I'd have thought. But it's in scanned it.
I've got it. Yeah.
And stuck it in there. So there's a range. It's from Dark Farm. Looked like it was a hop seller, which now goes to Mashed magazine or something like that. So I can't find the original, but the numbers are there, just as a kind of throughout the process. And the quote was from someone at Murphy and Sons at the top. Always check your pH throughout the brewing process, and then a list. So I kind of stuck to that a little bit, and it was
Yeah, it's interesting they've got the values for the liquor because well the trouble is with that, it's very difficult to measure your pH of something that's close to neutral. The slightest amount of anything will push it off because the logarithmic scale means that it's much tighter closer to neutral than it is at the far end. So
Okay.
if you measure distilled water, you can measure it like, I don't know, 3 times a day, the same sample, and it'll vary. You know, it's just because of temperature and the water will pick up, if it picks up any carbon dioxide, in other words, yeah, if it gets any CO2 near it, that's going to massively affect the pH. So anything with CO2 in it is going to be a problem measuring pH, which is why you'd want to remove the CO2 from it first.
Is that say because the logarithmic scale, it's like a kind of exponential curve, isn't it? Because it gets
Yeah, and it
further away,
yeah, exactly. So anything
or the other way
measuring anything
The other
close
way.
to 7 is tricky. You know, sort of like something will. I mean, it's a sort of theoretical point almost, neutrality, isn't it? It's it's
Like
where everything's
singularity.
balanced, where it's where your hydronium ions and your hydrogen ions are absolutely balanced, which is a sort of
Okay.
it's a moving point, you know.
I guess what we want from this little table is what does it mean if you're not in that range? And what do you do about it if it's not in that range? And how important is it?
Well, I'd say
that range is quite wide, isn't it? All the way down.
Well, that is it's like the
Fairly
water.
steep.
Your source water. It's not really going to matter what the pH of that is really at all. I mean, you know, measuring your source water pH isn't really useful in any way. So you know, it's once that water is part of the mash, then yeah, it's going to have a difference. And
I think you made a really good point earlier when you said it's literally the brewing process itself will get you in the ballpark in any case, depending
yeah, because
on your
people
water
spend a long
profile.
time. Yeah, I mean, it's like they're talking about the protein break and so on. Well, it's not a great surprise that it, you know, say like the best protein break isn't I don't know, a pH of 12 or something, because, you know, that would be pretty useless, if you see what I mean. You know, all of the figures, and they tend to follow the actual process. So it's dropping throughout the process, the pH, which suits the yeast nearer the end, suits protein coagulation, you know, before that and so on. So yeah, I mean, in a way, it's the process of brewing that's what the pH ends up as, sort of thing.
Yeah,
Because
it's
that's
just
how
kind of self-defining
how you
slightly, is
make
it?
beer.
Yeah.
Yeah.
Interesting.
I think the starting pH for yeast doesn't matter that much. Certain yeasts will always end up at the final pH because that's just what the yeast does, depending on the water and the buffer. But your starting pH doesn't really matter that much when it comes to fermentation because the yeast will get there anyway.
I was reading about something funny enough this afternoon about this, and they said, well, up to a point, because you don't want to stress your yeast. So they were suggesting that to get a good pH in your fermentation, do have a healthy pitch of yeast. Do make sure that the yeast isn't having to adjust the pH to make itself comfortable. Because as I said earlier on, the yeast needs to be at a higher pH than the external for it to be able to take in and out and do it all its exchange of all its all of the chemicals going in and out of the yeast cell. It needs to have a higher pH than its surroundings. So in order to get there, it's going to be expending energy and trying to get it there. So if it's there already, it's not going to have as much of a struggle. So apparently, if you can get your wort to a decent pH, it's less hard work for the yeast. Or if you're not going to do that, make sure you've got a really good, a healthy pitch of yeast, so the yeast doesn't have to do that adjustment at the beginning.
Or more cells, because then there's like a
Yeah.
bigger army working at it. Yeah.
Yeah.
Okay.
Yeah.
Yeah. Right. Hmm.
Yeah, because it was interesting to see that those that do the decoction mashes, that it tends to be a higher pH and then is lowered at the end. You're maintaining a highish pH, and that's where interesting when it mentions Maillard reactions occurring better at a higher pH. So keeping your mash pH, your pH high at certain points on purpose to create certain things.
Yeah, I've never heard of anyone doing that, but if you increase the pH in your decoction, that's probably a really good idea because you get more Maillard reactions, and the Maillard reaction itself creates more acidity and drops the pH again. So you drop it up at the start of decoction, and it itself will bring it back down again. And even so, because it's a logarithmic scale, that even when you add it back to your original mash, it's not going to move by much. Yeah.
Yeah, you've got that. I suppose because you have got that effect that in order to cause a difference, you're pushing against something, and because it's a logarithmic scale, the further away you get from neutral, the more you've got to chuck at it. I mean, you can notice that we're trying to acidify your mash. You start using X amount, and you think, oh, well, I need to get it a couple of points further lower, and you're adding considerably more because you're trying to push it further up this logarithmic scale.
Exactly.
So changing a pH from 7 to 6 is relatively easy. Changing it from 6 to 5 is consequently more. It's like increasing
10 times.
10 times more. Yeah, and it's not quite like that because of the buffering that goes on with the.
Does the moment of silence mean there are no more questions? Okay, Jerome.
Yeah.
Thank you very much. That was very, very helpful, and very informative and very well researched. So thank you.
Oh, thank
Yeah,
you.
that's great.
Thank you.
Very useful.
Thank you.
Thanks, Jerome.
Oh, cheers. Thank you.
Got a massive list of things now to check.
So who's got what on the brew?
And so, Jerome, do you know your?
Go on.
I was just going to say, do you notice with your very hoppy beers then that your final pH is very I can never work out if it's high or low, but yeah, up in the fives.
It's high-ish. I mean, I'm not. I mean, when I did actually read that Scott Janish article about it, and it's not a massive game changer. It's not like it's taking it a point or so above. It's a matter of a 0.1 or 0.2, well, 0.2 or something probably difference. It's noticeable. But again, I mean, I wonder with some of these things whether it's too much to worry about, because I think the idea of actually putting something, although Steve was saying the idea of actually altering the beer to see whether it is any better with a lower pH or a higher pH, I haven't done, which which I ought to in a way. You know, if I was that interested in it, what I should do is just get a dropping pipette with some phosphoric acid and sit there dosing beer samples and see whether, you know, in the same way that people talk about adding salt to beers. You know, that, you know, people quite happily add salt to their food, but, you know, you think, well, actually, I might prefer this beer if it was a tad more salty or whatever. But the idea of actually sitting there and dosing some beers and then trying to work out.
I think there's some mention of this in one of Gordon Strong's books about, you know, taking a measured sample of beer from the fermenter and diluting an acid with some, you know, essentially measuring how to doctor it to taste. And he kind of says there's no magic number, but it's just to taste. Obviously his palate is pretty well calibrated, but I guess it can be done. And then scaling, you know, what you need to add to the whole batch.
Yeah. And of course, when you get into this sort of thing, it sort of comes into personal taste almost, though. Then when you think, well, one person would say, oh, I think that's quite one thing, and it's just their taste buds and their personal things is that they think, oh, I could do with that being a bit sharper or whatever.
Well, I think Steve said he doctored them with multiple different levels and then tasted them later. So that could be another way to, if you're trying to dial in something that you brew often, you can dial it in for the next time, maybe.
I think it's especially noticeable with lagers and hoppy beers. I find it really lifts them, and it's noticeable. And quite often, if I'm not happy with a beer and I'll test the pH, and I'm like, oh, actually, that was a lot higher than probably ideal. And then you drop it down to, I don't know, 4 something or whatever, and the beer really pops, especially for those styles where either fruitiness or cleanness are, you know, part of the key.
Yeah, there seems to be an agreement that the lower pH will bring out a crispness and so on, but possibly at the cost of some of the maltiness or the from what I've read, you know, that you do lose a little bit of the maltiness by having, but then that might be something you want out of a beer. So
at that late stage, Ian, what is it that you add to it?
I use phosphoric because from lactic obviously has a lemony taste to it. And phosphoric is sort of one of the least tasting acids. And I just stick sometimes even a probe in the side of the glass and just titrate a tiny amount in there until I'm happy with until I've dropped the pH. I think when I was testing it, I was just dropping it and noticed about down to, it was out of the ballpark, and I took it into a point overall. But it was, the taste was, I was surprised actually. Sometimes you do those side by sides, and you're like, oh, I better not get these mixed up. And, you know, sort of, I find it slimmer to tell the difference, whereas this was very noticeable. And I think a very easy way of lifting beer if you think that isn't clean and it's got that sort of fruity poppiness to it. It can really make a big difference.
So which phosphoric is that? Is that the 70 or 80 or the 15? And for 20 liters, what's the ballpark amount? Are you thinking a couple of mils?
I've had 75% phosphoric. I had that for years. I'm into, and you really don't use much at that percentage. I guess some folk, I think I've read some folks say down in order to make has been okay. I thought it was interesting seeing there's clearly a whole chain of stuff goes on that's going to add up to, yeah, a lot in the finished product. Does anyone use acids to adjust, effectively to adjust the water to hit the right mash pH?
But apart from the AMS stuff, is that what you mean? CRS?
That's got other things in there as well, hasn't it? I'm not quite sure that's the same. But the reason why I ask is I've done it successfully with sparged batches, but then I tried some non-sparged batches, and my water is already a bit like RO basically, and so it doesn't take a tiny amount of acid will fling it off the chart, very quickly, alter it. But then I think there's so little in it that maybe this is what you were getting at, Jerome, that actually that reading means nothing, because then there's no buffering to it, because there was nothing in it in the first place. So it looks very acid. I think I've got down now to like 4.6. No, I can't remember where I've got down to, and the mash is still way too high on a no-sparge batch. So I don't know where I'm going to have to take it to, but it's a weird process.
Well, I started before I knew enough about this. I was measuring my sparge water pH before using it. I was like, and because I'd worked out I needed to drop not a lot, and of course now I realise why it's not a lot of. I think Bruin Water was saying something like, I don't know, 0.5 ml or something of phosphoric acid in the sparge water to get it down to the sort of below 6, which they recommend for tannin extraction or lack of tannin extraction. And I thought, well, I'll put in 0.5 ml, and it took it sort of like to about 6.2 or something. I thought, oh, I'll stick in 1 ml. And the next time it was way, but of course it doesn't really mean a lot because there's no buffering capacity for anything in there. Well, there's a bit because I wasn't using RO water, so obviously the salts, the carbonates and things are going to make a difference, and of course they do make a massive difference. So. But trying to work that out chemically as to, to drop that water with that content in it down to 6 is, it's tricky to get it right, But as you say, it doesn't really matter, if it come out at, I don't know, 5 or something, then it would just be, there was nothing in that water to buffer against the acid I was sticking in.
But doesn't that
I think I've got
give it an artificially lower figure? So you're thinking you're putting more acidified water in because it's not buffered.
Yeah. Well, because basically it doesn't take a lot to push a pH from 7 anywhere. It takes very little to push it anywhere. And then, of course, how accurate is your pH meter? Because if your pH meter's reading can run between, I don't know, say 4 and 8 or something, then to be able, when it gets to the midpoint, where we're around 7, it's just all over the place. Just because it was that logarithmic scale. So trying to measure anything close to, which is why most of the most interesting information about pH meters and electrodes is to do with people who do water testing, because those who are testing water all the time, they're in that narrow band of close to neutral, their equipment has to be really good. I mean, measuring the difference between pH of 1 and 2 is piss easy. Sorry, it's very easy. Whereas, you know, between 6 and 7 is a lot harder. So those people that make equipment for water testing are, I mean, that's where a lot of the information comes from. You know, a company called Xylem, for instance, who did a rather interesting article about electrodes and so on. Because
Yeah,
of course
just check my-
they're also trying to measure water, which could be I don't know, in the sewage works. So although it's close to 7, it'll be very difficult to measure because of the quality of what they're trying to measure. If you see what I mean.
So I've just double checked and I'm down to 4.2 for my no sparge water is what I've been taking, is what I need to take it down to next time to see if I can get the right mash reading. Which just seems mad.
So Ian, what salts do you add? Do you just, your tap water is incredibly soft, right? So there's like
Yes.
very little salts in there anyway.
Well,
So
bang some calcium carb. Well, bang all the stuffs that I've got in my water.
yeah,
You
that's
can
what I
have-
was thinking.
You can have some of that. I'll send you some.
We
So
could do a
if you
water
add some
swap instead of a beer
gypsum
swap
and
channel. We could do a water swap channel.
yeah, once
Bring
you
some
add
over.
in gypsum and Epsom salts and all of that, then it increases the buffer. Then it takes more acid to move the pH, and you'll probably find it's okay. But as it is, when you've got really soft water, what you start with doesn't matter, or the reading is meaningless.
Yeah.
Like Jerome says, yeah.
Yeah, if you start with RO water, then that figure you got at the beginning is, you know, forget it. You know, it's not really that useful. Because of
But-
course it's only when you start adding things to water that it gets interesting. But then what you add to it can make a huge effect on, you know, the acidity or alkalinity.
Okay. But my logic was this. So prior to, I mean, I used the phosphoric acid for other stuff anyway, but prior to trying to get my mash right with acidifying the mash and the sparge, I used Fraser's technique, the acidulated malt, which is absolutely bulletproof. It's such a good way of doing it. I mean, it's just, you can get that dialed in and it just, I was knocking out perfect mashes for that. It's really good. But
It's really good, isn't it?
But then I got to thinking, well, hold on a minute. It's just ground sprayed with acid.
Yeah.
So why don't I put the acid in the water? And I've got to hit, at some point, I've got to get the same effect. So that's where I've been aiming, but it's just not quite as easy as it looks.
It's much more it's much harder to hit it exactly, isn't it? I've done both ways, and I started doing it with like dropping lactic acid, measuring it by the 0.1 of a gram, and getting wildly different pHs. But then I started using acidulated malt, and it was way more accurate. It seemed, anyway.
Yeah. Yeah. It just, I've looked back and I was at the point where I'm like, oh, do I put 400 grams in or 450? And I can work out with the acidulated, you know, just from experience. I need a bit more on because it's slightly paler beer or whatever it is, and it just hits the right mash
really
pH every
does.
time. But
I found it so much more accurate than
being it with
pipetting.
But of course, some of that buffering capacity is from the malt itself, as well as the acid that it's soaked in or whatever.
That's a good point.
So it's been quite, I mean, unless you knew exactly, unless all malts are exactly the same, which I know they aren't, then, you know, I'd be saying, well, what is in this, the acidulated malt, you know, what, because the composition of that malt will make a difference.
Just pills, isn't it?
So it's a very light malt.
Base malt, isn't it?
Right, so it's a very light base malt, which is going to have it's not a good one for lowering your pH to start with. which is odd. I'd have thought, well, try and use something that's already going to drop your pH, if you see what I mean.
I think
But
all the
it's-
Germans were just trying to get around the brewing rule.
There is
So
that. Yeah, yeah.
spray
Yeah.
it. You know, they
Yeah,
spray
they make
and in fact-
themselves a rule and then try and break it.
and that means that they can then use it in their Helles right the way through to anything else, doesn't it? So you start with the blandest malt and spray that up. But I still think there must be a level of acidity, you know, however many mil of acid on that, or concentration, or however you want to measure it, you must be able to at some point get that same amount in your brewing.
Yeah.
And then add the grain.
I just, in the chat, I put a link into a Brewer's Friend calculator, and there you can put in your original water, what salts you add, and then you can sort of put in a target pH, and then you can either put grist color or even your actual malts, and then it'll sort of give you an estimate of where it thinks the mash pH is going to be. I did that probably about 10 times and measured it, and it was always quite close. So now I just use it and don't even bother measuring anymore. So you can start with RO water, that's just blank, or with tap water, and just input it and then just work from there. It's also got options for different waters, sparge water and mash water and so on. You could play around with it. Different calculators have different things, just like James said, and so on, but this is the one I use.
Okay.
It's a bit of effort every time, but yeah, I sort of do that a few days before brew day.
They've updated that actually, because I used to use that, and there's like much better diagrams and stuff at the bottom as well.
What's that?
Yeah, the old one
I
is
found
still
it really
available, but
accurate.
it's not so good. Yeah, that's right.
Which one's this? Which one's this?
Brewer's Friend. I just put it in the chat.
Right. Oh, right.
I'll have a go with
It's
that.
a good one. I remember it working really well. Yeah, their
So
tools.
I
are
like
good.
that one because it targets pH as well, and it'll give you your residual alkalinity, and at the bottom, sort of give you your final water, and you know, is that within the range of what you're trying to look for and all of that. How accurate is it? I don't know, but I guess I've never used Bruin water, and I know a lot of people use that, and that's probably better from what I've heard. But this is not too simple, not too complicated for what I do.
No, at the end of the day, you test out what pH is, don't you? And that's probably for the biggest, isn't it?
Yeah.
Very good. Right. Thank you very much, Jerome. That was excellent.
Well, thank you. Thank you.
Yeah, I'm probably also going to say goodnight.
Yeah, I need to jump.